Okay, perfect. So we'll get it started. Good morning, everyone. Today we have one of our presentations with Dr. Gude, and his guests, and I will hand it off to Dr. Pecchia to give a quick bio and intro for everyone. Oh, Dr. Pecchia, I don't think we can hear you. Oh. Sorry, we just got some audio.
If Greg is unable to talk, I will just give us a little introduction to get us going. It's our pleasure today to have Dr. Philip Askenazi here, who is a classmate of mine and a professor of medicine and pathology at Yale University. He's really succeeded well, and he's gotten into something called exosomes, which we're going to hear about today, which is very exciting. And we're very pleased he's able to share his research and his vision with us. Scott Reed is our Russian River poet. He'll start off on things. We were going to have our 5 med students give a presentation of bedside telemedicine, but our equipment isn't working. So we had to stop that. We ran across a problem up in Boise, Idaho. But I want to thank the 5 residents, actually med students, who've been on rotation with me and congratulate them on the matchings they made, because that's a very big thing. So congratulations to the 5 of you. And with that, Scott, how about a poem?
Thank you, Dr. Gude, and good morning, everyone. This morning, I'd like to present to the rounds the poem called Covid-19 by Joan Barano. To thank Dr. Pecchia for emailing me this poem sometime back, a month or so, and I actually found it quite well done and well written, and I thought that would be a good addition to our curriculum for the poetry and medicine series that I'm conducting with Dr. Gude and Dr. Pecchia. Just a word or two about the poem. I like the language in the poem, and I like the use of the metaphor. What the poem seems to do is combine the images of COVID-19, that little spiky little critter with those little protein rods on the sides of it that we've all become all too familiar with, combines the language and the image of that actual virus itself with the image of with images of the house, of, housing, and with images of the city. So the idea that the poem seems to blend so nicely together is what happens when one of these little viral creatures enters the house, which is, of course, the metaphor for the body, and makes a good example for us to study in poetry and medicine. So here is, and the other thing that just to note is that it appeared in the Journal of American Medical Association, which is edited by Rafael Campo. So he is quite a notable presenter and teacher himself. So COVID-19 by Joan Barano. They like the dark, wet places in the house to hide in, dividing, crowding out the prior tenants. Unasked, they plaster over walls, paste the rooms with stucco. O2 wants to get out but can't push through new sheetrock. Meanwhile, outside, the flow of traffic slows. Blood cells en route to the heart sputter. The city lights blink out until the whole power plant shuts down. Auxiliary generators brought in aren't enough. What's left isn't a metaphor. It's us. Thanks for listening, Dr. Gude.
Thank you very much. First of all, I'm going to make an urgent appeal for the Ukraine. If you look down there, you see that website. I'm in contact and have been active in World Rotary. It's the International Rotary Fellowship of Healthcare Professionals. We have a situation where money can be donated to the British Rotary, and it's sent to the Polish Rotary, and they're really on the border with Ukraine. And we will, with those monies, buy medical supplies, equipment, whatever they need. Now, the nice thing about this, it uses Rotary honesty and business skills to get money donated right there. I've donated to this. I recommend it. And it's a way of helping our colleagues in the Ukraine who are as brave as any people have ever been, standing up to really a terrible assault that was unnecessary, having to do with power and politics and Putin. But in any case, do consider donating to this. It really goes to where it should be effective. Now we're going to talk about COVID-19. Next slide, please. First of all, I want to say three things that are important. Vaccine, vaccine, vaccine. Vaccines were really discovered by Louis Pasteur, 1822, 1895, French chemist and microbiologist. He was not a physician. He invented the rabies, anthrax, and fowl collar vaccinations. And vaccines are the main prevention of a viral catastrophe. Let me give you an example. What was the population of the world in 1918? 1.5 billion. 500 million got the flu. That was the Spanish flu, or if you will, the pandemic of the influenza, and 50 million died. That was more than World War I by far, which was going on at the same time. We now have 9 billion people in the world, okay? And thus far, we've had 6 million deaths, 1 million in the United States. Back then, they had no vaccine in 1918, and they had no antibiotics. And of the 500 million cases of flu back then, there were 50 million deaths, 1 in 10. That is what will happen if we would get a variant of the COVID vaccine, COVID-19 type pandemic. If we would get a resistant organism that has to do not with treatment, it has to do with prevention, we would have the same problems they had back in 1918. And I'm going to give you a solution to that, I believe. The current population is 9 billion. 3 billion are the poorest. We need 6 to 9 billion vaccinations this summer to prevent getting something like would be a variant. It would be much more the Omicron or the Delta, but would be something like back in 1918. Without effective global, not US, US accounts for 5% of the population of the world, okay? That's all. Without effective global vaccination, more variants will likely develop over the next 6 to 12 months. One of them could be worse to or equal to the pandemic in 1918. It's possible. It's like in California, we're going to have an earthquake. Sure we are. It's not just possible, it's going to happen. Is there going to be an influenza virus come along that's going to be catastrophic? Sure there is. What is the effect of the current vaccine for influenza? How effective? 36%. Okay. Well, let's get off the flu and stick to coronavirus. The U.S. has developed 3 vaccines. It's a new technology. Pfizer, Moderna have the mRNA vaccine, Johnson & Johnson. They're very effective, 91%, 96%, 86%. But they're not the answer because they can't make enough and they're too expensive. Can't make enough and they're too expensive for the world. Next slide, please. Now, boosters work. They work for that 3rd shot. And right now, we're talking about a 4th shot. In Israel, they give a 4th shot to the elderly and those who are immunosuppressed. But the booster works. There's no question about it. Elderly, diabetes, obesity, immunosuppression, more available. That's just 99% effective preventing serious illness and death. Not getting infection, but serious illness, which means hospitalization and death. Effective from 5 years up. The answer to what we have are so-called vegan vaccines. Now, the reason you don't hear about these much is they're not patentable, okay? These are vaccines that are made by presenting a viral antigen to usually a recombinant strain of yeast, and you get an antigen manufactured. You use that in immunization. There are no human cells, no whole viruses, and no proteins used. These are what are used to make recombinant hepatitis B vaccine. We've got factories to make this in India, Bangladesh, Vietnam, Brazil, USA, and elsewhere. Vaccination costs $1.50 each, $1.50. The source of this information is the American Medical Association and a Dr. Peter Hotex, who's in the Baylor Medical Center. He recently got the AMA award for COVID research. You don't hear much about this because it's not patentable. And there isn't anybody backing this in terms of it's major use. But in India, they're making over 100 million of these a month right now. And that's just India itself. The answer is a cheap vaccine, and this is 90% effective, okay? And it can be used for booster. But the answer is Cobivax is what it's called. It's called a vegan vaccine. And so that is the answer. The answer to pandemics is Louis Pasteur's answer. It is vaccine, vaccine, vaccine. Okay, that's my main point. Next slide, please. The prevention, this is the Great Barrington Declaration. Look this up, if you will. This was a different approach to the COVID approach. It was done by the epidemiologists at Harvard, at Oxford, and at Stanford. And they would have taken a different approach of just taking the elderly and the immunosuppressed and quarantine them and leave the economy the way it was, schools the way they were. But we didn't take this approach. I signed early on, and I would have taken this approach. I was a minority. We took a different approach. Next slide. Now, this is the outpatient treatment. These slides are available to you. I'm going to talk about outpatient, talk about inpatient, talk about long haul syndrome, and then hand things over to my better colleague, Dr. Ashkenazi. Treatments not associated with any decrease in all-cause mortality. These don't work. Vitamin C, D, and zinc make no difference. Convalescent plasma, no difference. Azithromycin, no difference. Ivermectin, no difference. That parasitic class drug, the ivermectin. People still asking me for it in terms of coming to the hospital, particularly in Oregon and Idaho. But they're not associated with any decreased mortality. Treatments that did result in a decrease in the chances of hospitalization or death. Number one is an outpatient. If you get 3 days of IV remdesivir, 200, 100, and 100 IV, 3 days, that cuts down 87%. It cuts down going to the hospital or dying. Remdesivir works, but it's IV. And that's pretty invasive, if you will. The other thing that works is the molnupiravir, 5 days of that, 800 milligrams every 8 hours. It's an inducer RNA mutagenesis. It's a 30% lower risk, nothing like the remdesivir. Now, this is prevention, okay? Next slide, please. Now, this is outpatient treatment, and there's some new things that are on this. You've got the anti-spike monoclonal antibody, that's sotrovimir, 500 milligrams once a day, and that seems to be effective. Of the 3 available, that's the one that works, and it works with the Omicron. So that's the one that one would use if you can use an anti-spike monoclonal antibody. This is outpatient. It's really taking a person who's high risk and preventing them from getting bad illness. The other is using this combination that's above, but I don't use it because there's too many interferences with things like amiodarone, rifampin, rivaroxaban. I don't find it usable, but it does cut down the risk of death or hospitalization by 80%. Now, there are 2 new drugs out available. The one that works the best in terms of treating this in outpatient is the Pavlobid, and it cuts down the, if you take it as an outpatient treating COVID, it's a treatment, it's new, it was released in December, it cuts down the chances of getting hospitalization or death by about 80%. There's a second one, which is available, but it's only 30% cutting it down. So I won't espouse that. Next slide, please. And these slides are, and this is the hospital ward care once they come in the hospital. Number one, low flow oxygen. If you have to go to high flow oxygen, we use CPAP now. There's a good paper, JAMA, January. The CPAP is the best way to give oxygen over the high flow nasopharyngeal oxygen. We fully anticoagulate people who come in who are non-critical. That means they're not in the ICU. They're not on ventilators, and that is shown to decrease mortality. So we fully anticoagulate, we use CPAP for oxygen. Next slide, please. For delirium, we use IV dexmedetomidine, which is Precedex. The only thing that really works, the benzos and haldol don't seem to work. Seroquel and fluorophosphamine do help, but not as good as the IV drip. For sinus tachycardia, we use beta blockers, for hypertension, beta blockers, and alpha-1 blockers, namely labetalol. We favor famotidine over pantoprazole because it doesn't sterilize the stomach. And for acute kidney injury prevention, you really have to give fluids and avoid toxins like NSAIDs. And for diastolic CHF, it really is diuretics to diminish your tracheal edema. Next slide, please. Now, this is the complex ICU treatment of this. They all come in, they all get on dexamethasone. And remdesivir, dexamethasone cuts down the mortality by about 30%. Remdesivir cuts down the morbidity. It doesn't change mortality. And if they are requiring high flow oxygen or non-invasive ventilation, we add tocilizumab, which is an interleukin-6 blocker, or baricitinib, which is a kinase inhibitor having to do with inflammation. And if they really go further along, we use just dexamethasone, and we add generally tocilizumab as an interleukin-6 blocker. Now that's from up to date, and it's such a good diagram, I didn't do more than present it to you. Next slide, please. Just a quick rundown of how we treat this. And then we have the long haul syndrome. Now, it occurs in about 20-30% of the COVID patients, and Dr. Eskenazi will be talking about this particularly. The risk factors are probably these. Number one is the level of coronavirus early in the blood. So the quicker you get out of the blood, the thinking is that that will help prevent this. You get production of harmful autoantibodies. That's probably why the dexamethasone may work, and why the cytokine blockers may work. You reactivation Epstein-Barr virus, I think that's important. Diabetes is important, obesity is important. And the take-home recommendation is the quicker you can eliminate the virus, the less chance of developing COVID-19. Use remdesivir as early as possible because that gets rid of the virus. And try to treat the harmful antibodies. I'll defer to Philip on that. Control the glucose. Obesity you can't change quickly. And the role of exercise, sleep, diet, psychological well-being, all are being studied. And then you hear about the exosomes from my colleague, Dr. Eskenazi. So that is a quick rundown because I'm just a prelude. I want to hear what's new in terms of what can be done in terms of the long COVID syndrome. So Philip, I've run through mine rather quickly, so I want to listen to you rather than talk. I will defer to you, sir.
Well, Jim, you always were such a nice guy. And I don't know if the audience knows that we were medical students together. And at Yale, it's a very friendly situation, and we've maintained our relationship over the years. It's a pleasure to talk to this group about what can be called the long COVID syndromes, which are not getting really enough press now and are the sort of next pandemic, where something like 10 to 30% of people who have long COVID can end up with these post long COVID syndromes that are very complicated. One thing I would add to what you said is it's becoming clear that it's not necessarily the severity of the original COVID infection. And there are people who have not very severe or even subclinical COVID-19 that can evolve to have this syndrome, making it even more complicated. Now, I'm going to talk to you about some new ideas that have to do with exosomes, and I'll try to explain what they are. And they can be given by the nose and get into the brain where a lot is going on. And these are from mesenchymal stromal cells. So what are exosomes? They are nanovesicles released into the extracellular space. All cells release these, all species, all. That is down, all mammals, all biological fluids, all species down to bacteria and archaea. These are the universal particles of life. They're released from the endosomal pathway. They can be considered activated when the cell producing them is activated, and they then go to other cells nearby for paracrine effects. And all synapses that have been looked at so far have these vesicles, and I'll explain how they might be different. And then endocrine, they get into the bloodstream and then can affect things at a distance. This is a scanning EM. It's a defective, they are spheres, and they have a membrane distinctive signature, which leads to a certain receptivity of comparable ligands on cells. These are very tough, ancient universal vesicles of life. They're potentially involved in everything, biological and medical. I recommend you go to Google and put your favorite word in, let's say mast cell and exosomes or extracellular vesicle, and see what you get. They're in every subject. This is sort of an average diagram of the things they can have within them. Not every exosome has all these things. So they have certain adhesion molecules, ICAMs and integrins. They always have this set of CD that are markers of intracellular membranes. These are secreted intracellular and then leave the cell when it is activated and releases by compound sequential exocytosis. They can have MHC molecules. If they come from T cells, they can have the T cell receptor. One of the most important things they have in them is RNAs. A lot of RNAs, but we don't really understand what a lot of them are doing except the microRNAs that we do understand that are transferring genetic information between cells. The microRNAs work on RNAs to influence expression of genes. So that exosomes are extracellular vesicles that are the principal means of transferring functional genetic information between cells. We didn't know this was happening. They're universal vesicles of life. They're used by all species, as I mentioned. Now, with all cells releasing them, we have what I call an exosome cloud. So let's say this is an airway in an allergic kind of asthma sense, and all the different cells like mast cells and eosinophils and muscle cells and epithelial cells are all releasing these. Bacteria releasing what are called outer membrane vesicles, etc. The T cells and the presenting cells are all releasing to give you a cloud of exosomes. And clouds like this can be drawn from a neurodiagram in diabetes, exercise, cancer, etc. There are clouds to consider in all subjects. And this is what it's like on the ground. If you're a cell, this is what's coming at you. All of these. Now, we're used to white blood cells being at about 10,000 per cubic millimeter. In your blood, the exosomes are more like 1 billion. And you know, we just didn't know they were there. And there are billions of them. Now, the interaction between them is something like Twitter, an interactome like Twitter, so that a cell sends these messages out and is stimulated in a certain way to load up certain RNAs. And then they're received by another cell. And then that cell sends exosomes to other cells and so on. So you get primary, secondary, tertiary, as you might in Twitter and the conversations that go on. So that donor cells are broadcasting to all other cells, but they have a certain kind of barcode of adhesins, like a hashtag in Twitter, so that there's receptivity of certain cells compared to other cells for the particular vesicles and genetic messages. So what's so exciting about these things? They give us a new biological phenomenon and pathways to consider. They're the dominant, not only, but dominant mechanism of intercellular transfer of genetic function. They're universal, all cells, all species, down to bacteria, etc. They are likely involved in all biological and clinical processes. How involved they are, how crucial they are, remains to be determined, but they are involved. They bring new disease pathogenesis possibilities, or diagnosis is something called liquid biopsy, where a blood test focusing on the exosomes. I'll give you one example. The prostate makes these. They're called prostosomes. They're in the urine, and analyzing them is a much better test than the available ones. And then it opens up the possibility of natural nanovesicle therapy, which I'm going to talk about with respect to long COVID. So go to Google, put your favorite word in, see what you get with your favorite word and exosomes, and the more general term is extracellular vesicle. Have a look. So there's so many instances I could tell you of things that we know all about, but we didn't know the role of exosomes. So it turns out that mother's milk, or milk in general, is loaded with exosomes. And the mother is transferring not only IgA, but a whole lot of genetic information. And here are in the next slide. Hope we can get the next slide. Hello, hello. Somehow I got too bad here. Things have stopped.
Can you help out, Lauren, and take it from there?
Yeah, I can
Yeah,
share the slides if you'd like. Otherwise, you can try and stop sharing and then share it and then reshare, and it might work.
Okay, let's do that. And then share and see if we can reignite this. We better go over to yours, Lauren.
Is it still not? Yeah, I can share your slides from here if you'd like.
Yeah, please do that.
Okay.
You'll find this one in the middle.
Yep.
It's a great slide.
Yeah, there's a lot there.
The eyes.
So here are the exosomes in milk. So we don't know enough about this, but it seems that there are genetic messages coming from the mother to help the development of the neural system, endocrine system, immune system. I mean, in a neonate, the immune system's got to start being able to react to foreign things, but not react to its bacteria, not react to food allergens, is a very complicated situation. And the RNAs in the mother's milk are helping the baby develop these. Next slide, please. So now I'm going to switch over to mesenchymal stromal cells. It turns out the connective tissue of all the tissues, there's a cell we call the mesenchymal stromal cell. It's about 1 out of every 1,000 cells, a very rare cell. Next slide. But they can be grown up in vitro to 100%, from 1 out of 1,000, 1 out of 10,000, because they're adherent, they're self-replenishing, they're not phagocytic like macrophages would be, and they have surface markers that are quite different from what T cells, macrophages, and other cells would have. Next slide. So they're in the stroma of lots of tissues. The significance is that one gets them up to 100% and uses them in various injuries or models of diseases. They are healing. They're anti-inflammatory, they're immunosuppressive, I've mentioned about their phenotype and their self-renewal. So they have a non-T, non-myeloid, non-embryonic. They have a unique phenotype, and it's actually rather easy to grow them up to 100% and then inject them in various ways into disease models. Next slide. So this is a list of all of the disease models that you can find where mesenchymal stromal cells have been injected. Injury models, myocardial infarction, stroke, autoimmune models, pulmonary disease models, ARDS, COVID-19 pneumonia. And the significant thing is that the mesenchymal stromal cells can be replaced by the exosomes they make. And I'm going to show you an example of that. Next slide. So the more pertinent here about long COVID is that a bunch of long COVID has heavy neurological aspects. And the idea here is that maybe we could use them. They've been used for stroke. Models like Alzheimer's disease are being dealt with. Psychiatric disorders, neurotrauma, etc. So we are studying spinal cord injury, and we're beginning to give the mesenchymal stromal cell exosomes to patients with spinal cord injuries. Next slide. So the spinal cord injury is a major clinical problem militarily and civilian, high cost, premature death. In the laboratory, I don't do it, but they have a machine that injures the spinal cord of rats. They use 8 rats per group and treated 10 days after the injury with the mesenchymal stromal cells or their derived exosomes. And the animals can be analyzed in various ways to see the effect of the exosomes on the disease. Next slide. So if these are duplicates, if one just injects them every month, so here's a baseline spinal cord in a rat, and here's what it's like after the injury that is induced, and these are paralyzed rats. And if you give the mesenchymal stromal cells every month, you can get very good healing and regeneration of the various cells of the cord, including the neurons and the vascular cells. This is the effects of the mesenchymal stromal cells. Next slide. Now, a key finding is that if one labels them to try and understand what's happening, they mostly end up in the lung and the liver, and not a single one goes to the spinal cord, but the spinal cord gets better. So you inject these cells, none go to the spinal cord, but the spinal cord gets better. How could that be? It's because and we've shown this, that they end up in the lung and then give off their exosomes that go to the spinal cord. And I'm going to show you that. Next slide. So this is what they look like, the exosomes we get and the sizing of them. Here they're averaging about 80 nanometers across. And you go, this BB score is about locomotion, and you go from good locomotion to none. And then slowly it gets better on its own. But if you add the exosomes instead of the mesenchymal stem cells, you are improving and can keep improving. And of the many things we measure, the clearest thing here is in vascular permeability. You add a blue dye to the animals, and Um, with the injury, there is this permeability and increased blue dye in the tissues. And if you inject the mesenchymal stromal cells, you greatly reduce that. It has an effect on the vasculature, but their exosomes do the same thing. Next slide. And here is a fancy colored microscopy of this, and red inos is for M1 macrophages. Green CD206 is for M2 macrophages. There's this whole splay of different kinds of macrophages. And this DIR blue dye is the exosomes we've injected. And if we follow across this particular M2 cell, we see that it has rather specifically taken up the exosomes, so that there's targeting of this cell with quite extreme specificity. And it's a subpopulation of the exosomes and a subpopulation of the M2 cells that are involved. So we couldn't see the stem cells, but we can see their exosomes in the targeted cells. And next slide is a summary of our next paper in this as to what's happening. So the ones in the lung are giving off exosomes. We'll call those primary MSC exosomes, and they activate these M2 macrophages at the site of injury, not in general. And they give off TGF beta, which has some healing activity. And they also stimulate the MACs to make secondary exosomes with microRNAs that then influence the endothelial cells and the pericytes to make TGF beta receptors 1 and 2, and certain adhesion molecules that let endothelial cells interact with each other to restore vascular permeability. So you have mesenchymal stromal cell exosomes that target M2 macrophages that make some useful healing, and then produce local exosomes that influence the local vasculature to express receptors and occluding molecules that restore vascular barrier. Next slide, please. So that's a summary of what I just said. Next slide. So now we switch to long COVID and treatment with nasal exosomes. You may remember that at the top of the sinuses, there's something called the cribriform plate. So this is looking from below to the top of the sinuses. And these holes are where the axons of the first nerve, the nerve of smell, comes out at this dorsum of the sinuses. Next slide. And if exosomes are applied to the nose, they're small enough to be able to go through that space between the nerve and the skulls. The cells can't do it, but the exosomes can. And so this is the olfactory epithelium. This is the first nerve, and it's around the first nerve and to cells of the olfactory bulb. And so this is a way of directly administering exosomes to the brain. Next slide. Because they are physiological, they go through endothelial cells of the so-called blood-brain barrier. They actually can be shown to pass through the endothelial cells by something called transcytosis and make their way into the brain. And they are mainly taken up by macrophages and microglia. And we've seen how M2 macrophages can be very important in what we were doing with systemic exosomes, and spinal cord injury, this is now introducing the idea of nasal exosomes. Next slide. So here's an example of the model of multiple sclerosis called EAE. And the higher the number here, the more affected the animal is with this immunological multiple sclerosis. And various kinds of exosomes are given at the start here. And if they're loaded with certain inflammatories, then there is a tremendous reduction in the multiple sclerosis model. And intranasal exosomes have now been given to models of stroke, epilepsy, Alzheimer's, and even autism. So it's a new guy on the block. Exosomes are kind of new. The idea of giving them via the nose to pass the cribriform plate, to pass the blood-brain barrier, to get into the brain is kind of somewhat new thinking. Next slide. So here's a mouse model of Alzheimer's. Over on the left is a tremendous increase in the numbers and activation of microglia, and treating the mice with nasal mesenchymal stromal cell exosomes reduces that. There's a certain antigen that we're staining with. So, and other aspects of the mouse model as well. Next slide. Now, if we switch to acute COVID, so in the acute infection, there is brain involvement, and in both the gray matter and the white matter, staining with two different stains, you can see that there's activation and increase of the microglia as we saw in the Alzheimer entity. So this is acute infection. Next slide.
Phil, a question, that microglia reactivity, is that a virus or is that just inflammation, or what's the brain involvement?
There's activation of the neurons and of different cells, and it's not infection, it's some inherent and immunological thing. One of the strangest things that the neurons make eotaxin. And if you look in the CSF, there's very large amounts of eotaxin, but no eosinophils, which should be. So I'd say we don't really clearly know, but it isn't a virus, but it's some effect of the virus on the system, probably directly, and also directly on the vessels and activating these cells in ways that we're just beginning to know. There was just a terrific paper, I think in Science, in England, where they did CTs, and they had people that had CTs before they ever got COVID, and then CTs while they were acute, and they had very discernible changes in brain white and gray matter. So there is a big neuropathological aspect that is focusing on neurons and vessels. Next slide.
Thank you.
So now we come to long COVID. It's a very miserable, if any of you have seen these patients, it's something like chronic fatigue, but has some differences. Now, it's a very complex syndrome. There's more than 200 different symptoms in 10 different organs. The next slide shows the frequency of the symptoms. So this evolves a week or 2 weeks after the acute COVID, and involves 10 to 30% of the patients to some degree. The biggest thing is the fatigue. That is a huge fatigue, something like one would see in chronic fatigue syndrome or post-mononucleosis. This is post-exertional malaise, where and so on. But it has a lot of symptoms in a lot of organs. Next slide. Now we don't have a lot of brain material from patients because people don't die of this, and people don't want to have brain biopsies. But we do have some information showing a normal individual versus a long COVID patient, white matter and gray matter with activation of the microglia, like we were seeing in the Alzheimer's models and like one sees in the acute COVID patients that dies. So certainly one of the things is the microglia. And I mentioned that the nasal exosomes go principally to the microglia in the next slide, almost done. So I'm going to skip that one. Next slide. So conclusions about all of this. Long COVID often has dominant effects in the central nervous system. The patients end up in pulmonary clinics because there's such an emphasis on the acute pulmonary disease. But often the pulmonary doctors are having to deal with neurological syndromes. And at Yale, we now have a post-COVID clinic, both in the pulmonary department and the neurology department. In the COVID acutely, there is strong activation of microglia, like one sees in Alzheimer's, as well as vascular endotheliopathy involving microclots and decreased perfusion. And the proposal here is that given the fact that the mesenchymal stromal cell extracellular vesicle exosomes are helpful in certain disease models like stroke, like Alzheimer's, the proposal here is to do trials of the long COVID that might be susceptible to this therapy, which in the spinal cord instance induces healing TGF-beta and then secondary exosomes that affect the local vasculature through releasing microRNAs and changing DNA expression. So I'll stop there. I'd love to have questions if we have time for that, Jim.
Great. And hello,
Great.
everyone.
Take over.
Yeah. Thank you for that. Sorry for the muted state earlier in the presentation. Anyway, Dr. Ashkenazi, that was absolutely tremendous and eye-opening for all of us. I'll lead with a question regarding the evolution of the knowledge of clinical applications of MSCs, mesenchymal stem cells or stromal cells, and their derived exosomes. How does that square with the current research and applications that are going on for the use of stem cell therapy for neurologic and orthopedic syndromes in particular? Otherwise stated, are we seeing the evolution of a immunobiologic approach to the treatment of various diseases and conditions that include the direct deployment of genetically engineered exosomes produced at scale using the current supply chain that we now have in place for the production of mRNA-based coronavirus vaccines? So essentially leveraging the opportunity to quickly deploy the knowledge that you've just presented and get it out there to help those suffering from syndromes like long COVID.
My answer is yes, 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 and yes. That's a very complicated, sophisticated question. And yes, one can manipulate what's in the exosomes by manipulating the donor cells. First of all, they could be activated to be more, if you're dealing with microglia or macrophages as the source of the exosomes, to train them up with cytokines that would direct them in an M2. But also one can have them express genes that you've put in, and you also they're susceptible to CRISPR manipulations. So where you harvest them is one thing, and then just the natural ones versus all the things that between us, we mentioned a bunch of molecular things that one can do to them to give them certain properties. They have a tremendous advantage over giving the cells, because you're not giving cells, you're giving secreted vesicles. Cells, you're giving genes, and you have the problem of that they could become tumors or whatever. So this is a sort of a complicated reagent, a natural vesicle and they're far superior to the artificial forms. Like artificial vesicles cannot pass the blood-brain barrier, but natural vesicles can. Also, we have data of using them orally, where I just told you about mother's milk, where the exosomes in the milk are absorbed by the infant and cause molecular changes in the infant. And we have a system of immunology where we give T cell regulatory exosomes by mouth and can affect immunological reactions in the skin, say. So this is a new world, and depends on certain technical things of choosing how to make them, then making them to scale. But another nice thing about them is you can freeze dry them. You don't need minus 50 or 60, and you can add media. They're very, very tough old particles. So this is I mean, I've been in this field for 10 years, and the first international meeting had 25 people. Now they're like 5,000 people at international meetings, and they're in every field you could imagine. So look up and see what's happening in your field. Put your favorite word in, and I'd love to talk to you further. My email, Lauren can give you, and my phone number from Jim. I'd love to talk. It's a very sophisticated question.
Tremendous. Thank you for that. I'm sure we have some
So
additional.
I have a
minutes.
question.
Hi.
Mhm.
Hi.
Hello.
Good morning. I am Dr.
Hello,
Linda
Linda.
Gude. Do you
Yes.
hear me?
Yes, yes. Go on, please.
I would like to say thank you for this presentation. What kind of lab tests do you recommend to prevent this kind of sickles? So for example, the patient leaves the hospital, and after the hospitalization, we don't know what kind of consequences it's gonna have. So what kind of lab tests you can recommend to prevent that?
Well, I wish I could answer.
The CNS consequences.
That's a very important question, because you're dealing with a syndrome that is mainly symptoms. And it's very hard for us to deal with that. We're used to objective blood tests. And I say we don't have that yet, but there's a lot of development going on. I would say that in Israel and England and France and Germany are running ahead of the United States in the examination of this. The NIH money that was set aside for this is mainly going to epidemiology and not pathogenesis and not therapy, whereas European countries are already into understanding pathogenesis and beginning to work on therapy. So it's a very, very, very crucial question without a good answer, Linda.
Phil, a question about nasal administration.
Hmm.
You're an expert in that. You've cribriform plate. You've been thinking about it. Being cross on. Are vaccines any better given by nasal? I know some people think they are.
Oh yeah, Akiko Awasaki at Yale, who's our super COVID viral person, already has a paper out experimentally showing that nasal immunization is a very good way to be going for COVID itself. Yes.
Thank you.
I think we have time for one more question, and then I'll hand it over
Thank
to
you.
Lauren.
Thank
Wrap
you.
up.
Thank you.
Phil, I have a question. What's the best anti-inflammatory of this inflammatory disease?
Steroids.
Steroids, with all the complications.
Yeah, and that, well, that's a problem. You could say for these mesenchymal stromal cells. What about long term? So they're kind of like steroids, and we're giving them at very hyper physiological doses, and acute doses seems to be all right, but TGF beta is involved, and I think we still have to learn what's going to happen chronically by analogy with the steroids as well.
Thank you.
Lauren,
Please-
before I hand it off to you.
Please contact me via my email address, which I think you have in the handout, and I'd be glad to interact with any of you about any aspect of this. Thanks for having me.
Yeah, and it's, thank you so very much, Dr. Ashkenazi. We do have the email posted in the chat, and I did notice that Lauren had also posted a link for Ukraine donations. So Lauren, if you could comment on that a bit before you close, and thank you again to our speakers, our clinical speakers, as well as our poet in residence, Mr. Scott Reed. Thank you so very much. Lauren?
Yeah, so thank you everyone for joining today. I did post the link for the Ukraine donations, as Dr. Gude was talking about earlier today, in case anyone would like to donate to them, as well as Dr. Ashkenazi's email, in case anyone wants to reach out to him to directly discuss what he presented today. But other than that, thank you all for coming. It was a great presentation.
Lauren, I don't see that. Is it in the chat?
Which one? They should be both in the chat. Yes.
I don't see it on mine, dude. Does anybody else? The link to the Ukraine Rotary donation.
I can. Yeah,
It's in
I'll
the
resend
chat.
it. Yes.
It's in the chat.
It's
Do you
the
see it
first
now,
thing.
Dr. Falk?
Now I see it.
Okay. Perfect.
Very good. Thanks,
Perfect.
Lauren.
Of course. Thank you all for coming. Thank you to our presenters. It was a great morning, and I hope you all.
Thanks, Phil. Pleasure.
Thank you, Jim. Thanks, Lauren.
Of course.
Bye-bye.